Dear students, now I am going to tell you about antigens, antigenicity and MHC molecules. So, several times you are using the word antigens. So, what exactly antigens are? Antigens are substances, they are foreign substances, foreign protein, bacteria or viruses or pathogen or any macromolecules. So, when introduced into the body of a living animal, it evokes specific immune responses. What are specific immune responses? Either they induce the production of antibodies, specific antibodies, or they may activate T cell. So, these substances are called antigens. Antigens are defined as molecules that interact specifically with B cell receptors, BCR, or T cell receptors. If they interact with B cell receptors, they can able to induce humoral immunity or B cell mediated immunity through antibodies. If they able to bind or interact with the T cell receptor, they can induce the humoral cell mediated immunity or T cell mediated immunity. So, both these their interaction, it is carried out through major histocompatibility complexes. If it is complexed with MHC molecule, then only they can able to specifically interact with BCR or TCR. Antigen is usually protein in nature and sometimes polysaccharides. Majority antigens are protein in nature that generate specific immune response and induces the formation of specific antibody, or they induce the specific antibodies generation or production through B cells, or else they sensitize T cells. Sensitize T cells means they activate T cells, or else they do both the things. So, those are called antigens. Antigens are targeted by antibodies. You will see this. These are viral particles. They are these Y-shaped structure. This is antibody. Usually antibodies are, because this is the structure of the common antibody immunoglobulin G, IgG. That is why usually they will be picturized like this, Y shape you can see. See the Y shapes, these are all antibodies. So, how they are binding with the antigen. So, this may be a viral particle or else a pollen grain, whatever, or a fungal spore, all these. So, antigens are targeted by antibodies, are substances usually originated outside the body. So, they are originated outside the body. So, examples are viruses, bacteria, protozoa, substances produced by them, either viruses, bacteria, protozoa, or it may be snake venom. Snake venom is also a proteinaceous, it is a foreign protein. Certain proteins that is found in the food, they may be the toxic. So, they are originated from our diet, or components of the serum when it is get the blood transfusion. So, if it is there is a you can see the blood antigen compatibility is not there, then you can see those are the components from the serum. Antigenic proteins in the RBCs. So, other cells from other individuals, or else other animals, or else from the plants, or else from the fungi, all these are the foreign substances. They are getting to the body of an animal. They may induce humoral immune response or cell mediated immune response. So, those are called antigens. So, see here just this picture or the cartoon I taken from Lippincott pathophysiology. So, this is the macrophage. So, it is engulfing the antigen phagocytizing, breaking down into smaller components or peptides. So, immediately the MHC molecules, they bind with and they present on the surface of the cell. So, these are the they are presented. So, these antigenic peptides, when presented by the MHC, they can be able to sensitize or activate the T cells. So, the T cells, either they directly they may kill the antigen or inactivate the antigen, or else through stimulating the B cell to produce specific antibodies. This is how the antigens are presented on the surface of the macrophage by complexing with the MHC, major histocompatibility complex. So, antigens are immunogens. So, immunogen is a stimulus that produces humoral or cell mediated immune response. So, antigens are also called immunogens. They can able to induces the or evokes the antibody production, humoral immune response, or else cell mediated immune response. Cell mediated immune response, that is T cell mediated immune response. Antigens are any substance that binds specifically to an antibody. You can able to see they can able to bind with an antibody or else T cell receptor. So, all immunogens are antigens, but all antigens are not immunogens, because some small molecules are called haptens. So, these haptens can bind to antibodies or B cell receptor, but they cannot initiate the immune response. So, that's why the all antigens are not immunogens. So, these haptens, if they complex or they are called incomplete antigens, if they are carried by some protein complexes, they can able to function as a complete antigens. So, this you will able to see this is a bacterial cell. You will find on the surface of the bacterial cells there are so many antigenic epitopes. So, what are these antigenic epitopes? So, epitope, or that is the antibody binds to only a small portion of the antigen, which is called epitope. An epitope is the smallest unit of antigen known as antigenic determinant. It is called antigenic determinant. So, all these the green color you will see these are just the cartoon, you will see these are antigenic determinant. So, antibody can specifically bind on this, these are called antigenic epitopes or antigenic determinants. These are the part of the antigen that is recognized by an antibody or B cell receptor or T cell receptor. T cell receptor or antibody or B cell receptor can able to recognize a small portion of the antigenic the portion. It is called epitope or antigenic determinant. So, you will able to see this also a bacterial cell. If you imagine, you can see there is the different types of antigenic epitopes. So, different antibodies can recognize this, because antibody specifically they bind with the or recognize the specific antigenic determinant. So, an antigen has several or many different epitopes, you will able to see here, and reacts with many different types of antibodies, you can see. So, the triangular determinant, circular determinant, and you can see the square type. So, all these are they bind with different types of antibodies. The presence of multiple determinants in an antigen is referred as polyvalency. So, it is so many different types of antigenic determinants, and this is the called polyvalency of antigen. An epitope is about 5 to 6 amino acids in length. It is a small portion of antigenic molecule, or this is called a small peptide. It consists 5 to 6 amino acids. It is these epitopes are immunologically active regions of antigens. These are immunologically because these regions are recognized by the antibodies, that is why. So, what is the chemical nature of the antigens? Chemical nature of the antigens, as I said, they are vast majority immunogens are proteins. They may be pure proteins or glycoproteins or lipoproteins. Glycoproteins they associated with the glucose moiety or glycogen moiety. So the lipoproteins are lipid moiety, the protein associated with the lipid. Or else they may be polysaccharides or lipopolysaccharides are good immunogens. Nucleic acids are poor immunogens, or nucleoproteins are good immunogenic. Lipids are non-immunogenic. So based on this, you can see smaller amino acids, heptanes they are not immunogenic. Lipids, they are poorly immunogenic, and steroids. Carbohydrates, polysaccharides are immunogenic, or they can elicit immune response, whereas proteins are highly immunogenic, they can elicit. So depending on the immunogenicity, or depending on the antibody ability to produce antibodies, so these this is what you can see, the chemical nature of the antigens. So antigens types or immunogens types. So based on their origin, they are exogenous originated antigens, are bacteria, viruses, all these are exogenous originated antigens. They may be allergens, pollen grains or food antigens. All these enter the body through inhalation, either through the nasal passage, or else through the along with the food, or else they get injected. They circulate in the body, are trapped by antigen processing cells, as we have previously seen macrophages, how macrophage going to process this, degrade this, and they present on the cell surface. Even the dendritic cells, in previous video we studied about dendritic cells, they also processing these antigenic molecules, and they present this on the cell surface, that will be understood or they will be recognized by the T cells. Then endogenous originated antigens, those are the proteins found in the human cells, cytosols or cytoplasm of human cells. For example, tumor antigens, cancer cell antigens or tumor cell antigens produced by the tumor cells or cancer cells, or else viral infected cells, they also exhibit some viral proteins on their cell surface. So these are called the endogenous originated antigens. Then autoantigens. So sometimes organisms own antigens, they cannot considered as the self antigens. They can stimulate autoimmune reactions. So immune cells fail to recognize the antigens of its own antigens, its own body antigens. So this is, they cannot categorize self from non-self. So you can see that evoke humoral immunity or antibody generation against self antigens. So this is called autoantigens, or normal proteins recognized by the immune system of a patient suffering from specific and autoimmune diseases. There are number of autoimmune diseases. They are called celiac disease, inflammatory bowel disease, psoriasis, all these, these are result when the immune system fails to recognize its own body cells, antigens, that is leading to, they are called autoantigens, that is leading to autoimmune diseases, rheumatoid arthritis, all this you can see. Alloantigens. Alloantigens are antigens found in different members of the same species. For example, if the A person receive the blood from the B person, the B person RBC having the antigen B, it will be antigenic for the A person, and it evokes antigen antibody reactions. So that's how those are called the antigens. Those are present on the members of the same species. They are called alloantigens. These are different types of antigens. Now, what is antigenicity or immunogenicity? Antigenicity can be defined as the ability of a substance to induce antibody production. Immunogenicity is the ability of an immunogen to induce humoral or cell mediated immune response. The smallest unit of antigen is known as antigenic determinant. So, this can evoke the antibody production in the when it is introduced into the circulation of an animal. An antigen contains number of antigenic determinants. We have studied already antigenic epitopes. Antigens, when introduced into the body, are degraded by the macrophages, host phagocytes, into number of antigenic determinants or epitope, and they are displayed with the help of MHC, and that can be understood by the immune cells to produce or to bring specific immune response. This is how this is the ability of an antigen to evoke immune response called antigenicity or immunogenicity. What are the factors influencing this antigenicity or immunogenicity? Degree of foreignness. Degree of foreignness means if they are exogenous in origin, they have strong antigenic property. So, the antigens of closely related individuals are less antigenic than distantly related individuals. Antigenicity depends on the size of the molecules. We have studied already. Large molecules, large proteins are highly antigenic than the molecules of low molecular weight. They are less than 10,000 Daltons. So, the smaller molecules like amino acids or else simple sugars or simple lipids, all they are less antigenic or they are not antigenic at all. So, examples, large molecules like titannous toxide, egg albumins, thyroglobulins, all these are large proteins. They are highly antigenic. They are their size is more than 10,000 Daltons. So, small proteins like insulin, it is just 5.7 kilodalton or 5700 Dalton are either non-antigenic or weakly antigenic. Chemical nature, that we have studied, proteins are more antigenic than polysaccharides. Then physical nature, particulate antigens are more immunogenic than soluble antigenic molecules. Species specificity, tissues of all individuals in a particular species possess species specific antigens. So, that is why it is very difficult to, for the organ transplantation or tissue transplantation, because the tissues, they have their own tissue specific antigens. Similarly, organs also organ specific antigens, brain, kidney, lens proteins, all these they have their own organ specific antigens. That is why transplantation is not easy. Autoantigens, self antigens, we have already discussed. Certain circumstances, lens proteins, you can see they produce auto antibodies, like the cataracts will happen. Thyroglobulins, so they also leading to the pathogenic condition, they are behave like a autoantigens. Genetic factors, some substances are immunogenic in one species but not in the other species. Similarly, some substances are immunogenic in one individual but not in the other. So, genetic factors also influence the antigenicity. Then age also influence the immunogenicity. Very young and old have diminished immune response when compared to the young people. So, this is what you can see, the age also influence the antigenicity. Then dose of antigen, this is very important whenever dose fixation for an vaccine, So, you can see the immunogen can influence the dose of the immunogen can also influence the immunogenicity. Degradability, antigens can be easily phagocytized, are generally more immunogenic. the route of administration. So this is very important whenever there is a vaccine administration. Generally subcutaneous route is better, more antigenic, more antigenicity than intravenous, or else direct to the veins, or else to the intragastric routes. We will come to this more about this later when we are discussing about the vaccines. So then next is the last part of this video is the major histocompatibility complex, MHC. So the what are MHC? Major histocompatibility are the receptor protein, or they are present in the antigenic processing cells. Antigenic processing cells, here you will see the macrophage. So the macrophage having this major histocompatibility proteins. So the antigens are processed and processed into smaller molecules, and they are presented with the help of this MHC or major histocompatibility complex on the surface of this antigenic presenting cell. Okay. So this antigenic presenting cell membrane, so they are present, this is the major histocompatibility proteins or complex. They are antigen molecules, determinants are they are presented here. Can you see this? This will be recognized by the T cells. So MHC acts like a cell surface vessel. It's like a cell surface vessel holding or displaying the fragment of an antigen. So it is processed here by the antigen presenting cell, that is the macrophages, and this MHC, it is holding like a vesicle like, and it is holding this the antigenic epitope or determinant, and it is displaying or it is alerting T cell, see this antigenic substance is there in the system. So the T cells can engage with this molecular complex via T cell receptor. MHC molecules is to bind peptide fragments derived from the pathogens and display them on the cell surface for recognition by the T cells. These are coded by tightly linked genes for specific immune response, are found in all vertebrates. So they are present in all vertebrates. In human, this MHC is called Human Leucocyte Antigen HLA complex, and the genes of MHC are recognized by three classes. There are three classes of MHC or major histocompatibility complexes. So the MHC1, MHC2 and MHC3. So the MHC class 1 is a gene encodes for a glycoprotein molecule which expressed in the surface of all cells, all nucleated cells and the platelets. The major function of MHC is they bind with the antigenic peptide and present this to the cytotoxic cell. So the cytotoxic cell can able to identify the antigenic epitope presented by the MHC1. Immediately it bring the immune response. CD8, that is the cell surface molecule of the T cell, T8 cell, they can able to recognize this MHC1. What is MHC class 2? These are the glycoprotein molecules expressed, they are mainly by the macrophages. So this is what you can see, the MHC2, the macrophage, displaying this molecule, and or else dendritic cells, or else the B cells. The major function of the MHC2 is to bind peptide antigen and present on the CD4 T cells. CD4 T cells are the one, they actually activate the B cells to produce specific antibodies. Then MHC class 3, it is a diverse group of molecules. They are the one, these MHC molecules, they evoke, they alert or activate complement activation and involved in the inflammation caused by the cytokines and tumor necrosis factors and etc. So this is how exactly, this is the antigen presenting cell or the macrophages. These are the MHC molecule, the MHC1 major histocompatibility complex 1, and this is the 2. So, the 2 will be recognized by the T cell CD4 T cells or helper T cells. MHC class 1 recognized by the cytotoxic T cells. This is how exactly, this is the antigen presenting cell, and the T cell receptor which it will recognize MHC holding the antigenic the peptide, and immediately this is the cytotoxic T cell, it neutralize or inactivate the antigen. This MHC2 it bind with the T4 cells or CD4 cells, and they can bring the humoral immune response or antibody mediated immune response. So, this is a cartoon, you can see how exactly the MHC will react. So, the MHC1 or human leukocyte antigen presenting cell, this one MHC1. So, it is whole protein will be presented to this is the cytotoxic lymphocyte or T lymphocytes. This MHC it is presenting bringing different parts, this is presenting further, it is peptides. So, this cartoon indicate the functions of the MHC1, MHC2 and MHC3. Thank you.